Good morning. My name is Mauricio Diaz Granados, and I am Chief Science Officer and Dean of the International Plant Science Center at the New York Botanical Garden. It is my pleasure to welcome you to the 6th Annual New York City Ecoflora Conference. This conference is being recorded, and in a few days it will be available to watch at a link that we are placing in the chat box, along with the archived recordings of the previous 5 annual conferences. First, I would like to begin by acknowledging that the New York Botanical Garden is located in the ancestral homeland of the Lenape Delaware people. We honor them and acknowledge their displacement, dispossession, and continued presence. The theme for this year's conference is the future of New York's city flora, threats and opportunities. My colleague, Dr. Larry Kelly, will introduce and describe the conference program in a moment. But before, I would like to briefly give you, to put in context, the New York City Ecoflora project in our institutional context. In a real sense, the Ecoflora project relies on the expertise of dozens of PhD scientists, technicians, and support staff who brilliantly curated the collections and database resources and conduct cutting-edge research on plants and fungi within the International Plant Science Center. And this includes the William and Linda Steer Herbarium, the CV Star Virtual Herbarium, the Lester T. Mertz Library, the Institute of Systematic Botany, the Institute of Economic Botany, and the Plant Research Laboratory, within which we have the Louis B. Dorothy Kuhlman Program for Molecular Systematics, the Genomics Program, the Structural Botany Program, Climate Solutions, and the Laboratory Collections. The Ecoflora project is also enriched by the International Plant Science Center's Commodore Matthew Perry Graduate Studies Program, conducted jointly with 6 universities. In fact, one of today's speakers, Lydia Paradiso, is a doctoral candidate in NYBG's joint PhD program with the City University of New York and has been importantly assisting with the Ecoflora project for the past 3 years. Another one of today's speakers, Dr. Eric Sanderson, holds the newly established position of Vice President for Urban Conservation, which is highly relevant to the Ecoflora project concept. Above all, the key elements of the Ecoflora project are the community scientists who volunteer to document the flora, fauna, and fungi in New York City. Community scientists are the real heroes of the Ecoflora project. As of today, more than 35,000 people have made more than 1 million observations on more than 12,500 species for the New York City Ecoflora project. Community science projects have become extremely important in addressing some of the most pressing research questions within the context of the intertwined biodiversity and climate change crisis. At NYBG, there are not just opportunities to engage with the Ecoflora project, but there are 2 other community science projects. The Herbarium Specimens Digitization, which aims to accelerate the pace of biodiversity by discovering and helping to digitize all 7.8 million specimens of plants and fungi in the William and Linda Steer Herbarium, and Phenology, observing and collecting data to study the effects of weather and climate change on plants in the family theme forest and in catalog and curated living collections on NYBG's 250-acre campus. If you are interested, please see the link that we are putting into the chat for connecting with NYBG's Volunteer Services Department about community science projects. Thank you very much for joining us for today's conference. I also want to thank the many community scientists who have contributed so importantly to the Ecoflora project to date, and for those who may become inspired to do so after participating in this conference. I now turn the program over to my colleague, Dr. Larry Kelly, who will introduce and moderate the conference. Thank you very much.
Thank you, Mauricio. Good morning. I'm Lawrence Kelly, Vice President of Science Administration here at the New York Botanical Garden. The topic of this year's EcoFlora Conference goes to the core of what the project is about: fostering a sustainable future for New York City's plants and their ecological partners, including humans. We have an excellent lineup of speakers this morning to address different aspects of this theme, but before I introduce them, I'd like to quickly summarize the EcoFlora project for those of you who are new to it. The New York Botanical Garden created the EcoFlora in 2017 with a grant from the Institute of Museum and Library Services, building on the garden's historical strengths in plant collections with emerging opportunities in digital technology. The EcoFlora, as Mauricio mentioned, is designed as a community science project to document the native and naturalized flora and associated animals and fungi of New York City. As a primary way to foster community scientist engagement, each month we sponsor an EcoQuest challenge, encouraging New Yorkers to observe one or more species, then upload their observations to iNaturalist. We're posting to the chat a link to our current EcoQuest challenge webpage, including the monthly challenges since 2017. The current month's challenge focuses on a recently arrived invasive insect, the spotted lanternfly, that has quickly become a significant threat to many ecologically, economically, and culturally significant plants such as grapevines, stone fruit trees, and maples. Community scientists are the human engines that power the New York City EcoFlora project, and we would like to take this opportunity to thank and honor the more than 35,000 individuals who have made observations for the project by recognizing some of the most prolific contributions over the years. The slide on the screen identifies the most active contributors to the project, who are highlighted not only for their own considerable efforts, but also as proxies for the collective contributions of community scientists to the EcoFlora. Congratulations and thank you to all contributors. For the past 4 years, New York Botanical Gardens EcoFlora project has continued with renewed support from a national leadership grant from the Institute of Museum and Library Services, with the goal to scale up and expand the EcoFlora model nationally in partnership with 4 other public botanical gardens. This slide shows the homepage for the portals of the EcoFlora projects at NYBG and our 4 partner public gardens, namely Denver Botanic Gardens, Chicago Botanic Garden, Desert Botanical Garden in Phoenix, and Marie Selby Botanical Gardens in Sarasota, Florida. On behalf of NYBG, I want to express our deep gratitude for the partnership we have shared with these gardens in expanding the EcoFlora model. If you'd like more background on the EcoFlora concept and a toolkit that the partnership developed for starting an EcoFlora project in your town, we're putting a link in the chat to a 2022 article in Public Garden magazine entitled EcoFloras: Fostering Community and Biodiversity Discovery. So let us now turn to today's 5 presenters. Each presentation will be 15 minutes long, and at the conclusion of all the presentations, we'll have time for a question and answer session with the speakers. As you listen to the presentations, if you have a question you would like to have asked to one or more of the speakers, please post it using the Q&A module, and these will be asked of the speakers during the concluding conference session. The first presentation is by Dr. Mila Aronson, whose research focuses on the patterns, ecological, and environmental and social drivers of biodiversity in urban landscapes, in particular, understanding how to incorporate nature into our world's cities. She received her bachelor's degree in natural resources from Cornell University and a master's and PhD in ecology and evolution from Rutgers, where Mila is currently an assistant professor in the Department of Ecology, Evolution, and Natural Resources, and the director of the Hutchison Memorial Forest. Dr. Aronson has used results from her research to direct decisions for city planning and design, restoration, and management of urban green spaces. She serves or has served on the scientific advisory committees, the Silicon Valley Urban Greening Panel, the Stewardship Mapping and Assessment Project in New York City, and Fire Island National Seashore. We now turn to her presentation entitled Biodiversity in a Concrete Jungle, Global to Local Drivers of Floras Across the World Cities. Welcome, Mila. I will now turn it over to you.
Thank you very much for inviting me to speak today, and I'm very excited to be part of this symposium. Today, I'm going to talk a little bit about global to local drivers, or what factors influence biodiversity, and in particular focused on plant biodiversity across the world's cities and in the Northeast in particular. So cities, our world is highly urbanized. As we know, as a species, urban people live in approximately 58% of the human population lives in urban areas, and we have but in only about 3% of the terrestrial land surface is urbanized. We're an increasingly connected, globalized society in that we move species and people around the world. I like to show this slide in the beginning of my talks because it really gets at how connected we really are as a global society. Across the oceans, we see lines across those represent shipping lane densities. And then on the terrestrial land surfaces here, this is the travel time to major cities of 50,000 or more, from less than an hour to all the way up to 10 days. And so the darker colors represent longer time periods and the lighter colors represent more. But I like to look at this and think about our cities and how we're moving species and people around, and we're increasingly connected. So why should we care about nature in the concrete jungle? Cities are disproportionately placed or built within areas of high biodiversity. Therefore, cities actually have quite an impact on global biodiversity and the global biodiversity crisis that we're now facing. Cities are growing rapidly. It's, as I mentioned before, over 56% of the world's population lives in cities, and that's about 83 to 85% in the US. Nature in cities is important for our health and well-being, both physical and mental health, as well as the services or ecosystem services that green spaces or nature offers to people, such as temperature, air pollution control, flooding retention, and so on. Nature in cities also is important for the continued resilience in the face of climate change and other social pressures and changes. So I'd like to just quickly go over the global patterns of urban biodiversity. So this is looking at the worldwide scale. This was a study that myself and colleagues performed in 2015, in which we collected databases from 147 cities across the world, across all the biogeographic realms, and we examined the birds and plant species of each city. These were birds included standardized surveys during the breeding system since 1990, and plants included surveys of natural and spontaneous vegetation since 1975. And what we found, so we were interested in what does biodiversity look like in cities? This was the largest database to date at that time on the flora and fauna of cities across the world. So just quick overview of what we found. We actually found quite a bit. We found that cities are not the concrete jungles that they're often assumed to be, as I'm speaking to the choir in this seminar here. These cities supported about 20%. So these only 54 cities in our database here supported 20% of the world's bird species. That was over 2,000 birds, and about 5% of the world's plants. There's a lot more plant species in the world. We found that cities are these novel ecosystems, as we assumed, with a mix of native and non-native species. So on average, cities supported about 94%. The bird species found in cities were approximately 94% native and 70% native plant species. but we did find few species of cosmopolitan. So we have this assumption that all cities are the same. They are very similar in their built structures across the world, impervious surfaces, housing, buildings. So we have this assumption that the flora and fauna of cities are approximately the same across, but we found very few species to be cosmopolitan. Of course, pigeons are found all over the world, and in this case, in over 80% of the cities. And for plants, we found only 11 species were found on all biogeographic in cities on all biogeographic realms. The most common plant was Poa annua, which is the annual meadowgrass. So we looked at these kind of questions about what types of species, particularly plants, that we were finding in cities in terms of these as metrics for understanding how cities are supporting global biodiversity. And we found that, I mentioned this just now before, that on average, cities supported, the floras of cities were approximately 70% native. And that held true throughout the different biogeographic realms that we studied. We studied the Near Arctic, which are cities in North America, Paleoarctic, which are cities in Europe and Asia, as well as the Afrotropic, so African cities, Indo-Malayan cities, and Australasia, so New Zealand and Australian cities. What we found was that throughout most of the biogeographic realms, including New York here, which is approximately has about, the flora of New York is approximately 68% native species. We found that different cities across the different biogeographic realms held this pattern, except for cities in Australasia. So for example, Hamilton, New Zealand is only approximately 48% native. This really represents how invasive they are in terms of invasive plant species, and how global geographic change or human-mediated biotic interchange, meaning how connected we are, has really brought species around the world and influenced and affected the floras of particular cities, such as in Australasia, more than others. So I always like to talk about what are the most common plants. I think this is really exciting. These are the plants that are found everywhere. These are what I call our city slickers, right? They're found in cities. We could go outside right now and find these plants within a 10-minute walk of pretty much wherever we are, right? But we have Poa annua here, which is the annual meadowgrass. We have Phragmites down on the bottom right, which is a giant reedgrass, an invasive plant. A lot of these species are what we would consider weeds, but they actually are quite adapted to urban environments. They're found, like I said, all across the world, in cities all across the world. So one of the other questions we asked about this study was what are the important drivers of biodiversity? So they're really common. We know that there are common drivers of biodiversity. We find more, for example, latitude impacts how many species we find across the world. So where there are more species at the equator, and then we have less species as you go north or south in latitude. However, we also know that climate affects biodiversity, how many species we find, but we are interested in the anthropogenic features. And if cities really do have a lot of commonalities. So are there particular aspects of cities that influence biodiversity more than your kind of traditional influences like climate and latitude? What we found, and this seems intuitive, but it was one of the first studies to really show this across the world, is that cities with more natural or semi-natural, including restored vegetation, like this is a restored salt marsh in Secaucus, New Jersey, right across the river from Manhattan. The cities with more natural vegetation support more biodiversity, in particular support more plant biodiversity, but also support more bird biodiversity, which is relying on those green spaces that where these plants are found in. And so that brings me to a focus on urban green spaces. We define urban green spaces as all natural, semi-natural, and artificial ecological systems within a city, and they comprise a range of habitat types from remnant natural areas such as forests found in places like Inwood Hill Park, all the way to home gardens and green roofs. And they are forests, urban green spaces are also managed differently. So they have different levels of management intensity and economic input, such as personnel, nutrients, and so on, pesticides, nutrients added, but as well as they exist along a gradient of native species diversity, where you would find most native species within more natural to semi-natural areas, such as the riparian corridor in this example, or in remnant natural areas. And so a lot of the work that I do really focuses on the global scale, but also focuses on what within these urban green spaces is influencing biodiversity. And because if we understand that, then we can better plan, manage, and design for biodiversity in our cities. And I focus on urban green spaces because not just because they are the places where we find most species in cities, right? They support ecological communities, including rare and endangered species, as the EcoFlora project has found. They also support ecosystem functions, such as this park, Tanner Springs Park in Portland, Oregon, which serves as a green infrastructure or for stormwater retention. So they support ecosystem functions that provide a wide range of these ecosystem services that we rely on as city inhabitants. But they're also the nexus of social ecological processes. Green spaces shape the character of the neighbor and its neighborhood. They provide a sense of place for urban residents, and they provide a range. There's a lot of work that research that has found that urban green spaces provide a range of social, physical, and psychological health benefits. We all feel better when we go out in nature. The Japanese have coined this forest bathing or nature bathing. And these social ecological systems, if they're functioning well to support both biodiversity and people, they add to the resiliency of the city itself, of the landscape, in the face of climate change and other large storm events and other heat waves and other social changes that occur across cities. So we've looked at I'm just going to show you a couple of factors that are really important in shaping urban plant communities. One of them is competition with invasive plant species. Some of the work that I've done has shown that competition with invasive plant species reduces canopy tree regeneration. The canopy tree, and in this study, we looked at canopy seedlings in tree seedlings, and those are the next generation of trees. So we're really looking at how we can, I'm really looking at how do we understand the factors that determine the next generation of trees so that we have a forest in our future. And what we found in this study in particular was that when seedlings were planted or grown within this grass, Japanese stiltgrass, Microstygium viminium, they not only survived less, so we had lower survival, but also lower of those that did survive, we had lower growth. And so we do need to manage these invasive plant species, such as Microstygium viminium, in order to promote the next generation of trees. I have a former graduate student who studied seed predation in urban and rural forests. So we had 3 urban forests, which were here in New York City, in Van Cortlandt Park, Pelham Bay Park, and Inwood Hill, as well as 3 non-urban or more rural, more contiguous forests in southern New York, upstate or southern New York State, depending on your perception. And what he found was that he set out seed traps. So he set out seeds for to be predated upon, to be eaten by rodents and birds and whomever. And what he found across 3 years, so in this graphic, we're looking at the red lines indicate the urban sites, those forests in New York City, and the black lines represent the rural sites in southern New York State. And what he found is that across time, urban forests had elevated but sustained rates of seed predation. And while we see the cyclical cycle where we have high seed predation in the rural sites in 2016, low seed predation in 2017, and then high seed predation again in 2018. And so this indicates that something is different in the urban sites in terms of the granivore or the seed predators. And so it could mean that the seed predators, there's higher levels of seed predators, more squirrels, more chipmunks, maybe in the urban areas. There could be different seed predators, so more rats, for example, in urban than rural areas. But either way, urban areas experience higher elevated seed predation, showing that this is also not just competition, but with invasive plant species, but also seed predation is an important aspect driving or influencing plant communities. And so using all this knowledge, using all these studies and all this knowledge that's coming out of what is influencing at both global, regional, and local scales, what influences plant communities or the plants that we find in our cities, we can use this to then build biodiversity, to restore, to conserve, and preserve biodiversity into our cities. I worked with the San Francisco Estuary Institute, and I advised them in this publication called Making Nature City, in which they took a lot of the knowledge that's been generated in the last 10, 15 years and put it together in terms of actual recommendations for building and managing biodiversity in cities. And so a lot of this has to do with, so they came up with 7 main themes. Patches, so having larger patch size, there's a lot of work that shows that larger patches are important, but as well as these small patches of native vegetation are important in supporting biodiversity. Putting in connections amongst habitat types so that we can connect plants and animals move throughout the city. Plants move via their seeds, right? And so having connections that allow for movement of plants and animals to move to new places, made the habitat diversity, so having lots of different, so New York City in particular is very diverse in forests and coastal salt marshes, freshwater marshes, grasslands. We have a lot, and urban grasslands, urban green roofs. All of these different habitats are important in maintaining different biodiversity across the city. Also supporting special resources such as large trees, wetlands, streams, and rivers in riparian areas along the rivers. And then management, both in terms of restoring and planting native vegetation in all types of green spaces, whether that be Washington Square Park or Pelham Bay Park. Increasing the amount of native vegetation will also increase higher trophic levels, such as our pollinators, our birds, and so on. And then management, not just planting, but also managing for biodiversity in our cities to increase higher biodiversity outcomes. And so more specifically, I want to talk about this go back to the different scales, right? So at the citywide scale, we can increase naturally vegetated green space, right? Most of our cities, especially if we're talking about New York City, a lot of our cities are built, right? So there's not a lot of there's not area to make new huge natural parks, but we can increase the native species biodiversity of our existing parks and increase other naturally vegetated green space, small green spaces. And then also at the citywide scale, increasing connectivity within the city as well as within the surrounding region. We can, at the neighborhood scale, fostering connections among residential yards, for example, because a lot of our cities, including New York, has residential yards. If you look at Queens, a lot of the green space in Queens is residential yards. So how do we utilize people's yards and gardens to foster connections among green spaces? How do we use yards and gardens in Queens to foster connections or stepping stones for pollinators, for plant species to move from the Jamaica Bay National Wildlife Refuge into the city, and as well as from the city into the wildlife refuge? And of course, community engagement. None of this can happen. None of this restoration or building biodiversity or design can happen without community engagement. So involving people and into caring about biodiversity, as the EcoFlora project really excels at, but also in terms of actual actions for restoration, for planting in their yards and gardens. And then of course, at the habitat scale, thinking about the soil. So one of the things I didn't talk about is the soil and how important the soil is in soil health for plant biodiversity. And therefore, and then plant biodiversity is important for insect and bird biodiversity. Increasing vegetation structure. So one of the things that Thane Family Forest is really focused on in the management is increasing the vegetation structure. So adding herbs, shrubs, trees, having all of these strata. This graphic on the bottom shows the estimates of bird biodiversity from 1980 to 2005 for 21 forest breeding bird species in New Jersey. And so anything above the line means those bird species are increasing from that time period. Anything below the zero line indicates that they're decreasing. And so what we found really is that the shrub and ground species, and even in the mid-story, those species are decreasing. And that's because of this decrease in shrubs. In our cities, a lot of people perceive a lot of vegetation as messy, or they perceive it as dangerous, that criminals could be hiding behind the shrubs. There's no actual trends to show that that's true. So we need to increase our shrub diversity to then and volume to increase biodiversity, planting native plants, and of course, invasive species management. And with that, I'd like to acknowledge all of my many collaborations and coworkers, and I will take questions at the end.
Thank you very much for that perspective on global and local drivers of plant diversity in the world's concrete jungles. Really appreciate it. Excellent presentation. Next up, we have a presentation from Lydia Paradiso, who's a PhD candidate in the joint program between the New York Botanical Garden and the Graduate Center of the City University of New York. Lydia's research focuses on the current and historical spontaneous flora of New York City, as well as systematics of the conifer genus Larix. Lydia is also the president of the Torrey Botanical Society, which is the oldest botanical society in the Americas. Lydia received a master's degree in biodiversity and taxonomy of plants from the University of Edinburgh, and before that, she got a bachelor's degree in bioinformatics from the New Jersey Institute of Technology. Lydia is going to talk to us today about the spontaneous flora of New York City. Welcome, Lydia, and I will turn it over to you.
Thanks, Larry. All right, let me share my screen. All right. So like Larry said, I'm currently a PhD student at the New York Botanical Garden, and I've been here just over 6 years, but for the last 3, I've been working with the EcoFlora project. And one of the big goals that we had for this project was to use all of the data available to us to compile a checklist of the spontaneous flora of New York City. So that's what I'm going to talk to you about today. So really quick, what we all know many reasons that make New York special, but there are a couple of specific kind of ecological, geological, and climatic related things that really make it unique. So for example, the geology of the area is really interesting. Along here, this line, this orange, is the terminal moraine, which during the last glacial maximum about 10,000 years ago, that was the line where the glaciers came down to. So everything north of that was covered, was glaciated, and everything below that was not. So that really shapes kind of the assemblage of plants that we'll find today, as well as the substrate, et cetera, that we find as well. Also, a lot of sediment was deposited by these glaciers. So we get some really interesting deposits in Staten Island in particular that we don't find in the whole rest of the city. We also are at the crux of 3 different level 3 ecoregions. I'm trying to remember the names. There's the Piedmont, the Atlantic coastal pine barrens, and then the third one, whose name I don't recall. But we're at this really interesting confluence of these 3 ecotypes, and it's one of the world's largest natural harbors as well. Looking at some other things going on here, on the left we have the historic tidal and freshwater wetlands, as well as what we have today. So in purple are the historic areas, and in green are the current areas. There's been a loss of over 90% of both the tidal and freshwater wetlands in the past 100 years. But there are still some amounts, quite a bit, that remains. If you look on the right, we have this awesome map from the Natural Areas Conservancy that if you go on their website, they have an interactive feature that you can see by borough, by park, how much land is green space, how much of that is wild green space, and what we have of these saltwater and freshwater wetlands. So even though we can see on one hand, there are a lot of the kind of fragmented spots, there's a lot of areas where there is no kind of wild green space. There are quite a bit of places where there are. And so these are places that harbor tons of really interesting biodiversity, but these are also places that are at risk for lots of things with things to do with climate change, with sea level rise, but also with development as well. So the things that we're asking are, what is the current composition of the city's flora? What changes have occurred in the last approximately 200 years? And what traits are associated with persistence or loss? So really quick, crash course of botany in New York City. The first checklist of plants in New York City was in 1811 that covered the island of Manhattan. That was followed a few years later by John Torrey's catalog of plants growing spontaneously within 30 miles of the city of New York, which is one of the data points that I used for the historical comparison. The Torrey Botanical Society was founded about 50 years later by followers of John Torrey. They did lots and lots of work on the flora of New York. For example, this 33 miles of New York City checklist that was published over several years by William Leggett. The New York Botanical Garden was founded in the late 1800s by members of the Torrey Botanical Society. The Brooklyn Botanic Garden was founded not soon after that. And then in the early 1900s, we get this book by Nathan Taylor, 100 Miles of New York City Checklist. That's my second data point that I'm using. In the interim, now in the 1900s, there wasn't a ton going on in the New York City flora specifically until the 1990s, when the Metropolitan Flora Project was started at Brooklyn Botanical Garden, which looked at current and past distributions of species in this large kind of area, similar to the 100 miles that Nathan Taylor looked at. We had a checklist in 2004 by DeCandido et al. That looked also at the current and past species. and something that was produced by, oh, and then we have the New York City Ecoflora. So that's where we come in, collecting data using iNaturalist to supplement all of this other stuff that has been done before, to see how things have been changing and what we can additionally find, or what we can find has been lost. So really quick, we talked about, I'm mentioning spontaneous plants a lot, but what is a spontaneous plant? So a spontaneous plant is something that's not planted by humans and is persisting. So for example, you'll find, if you find a dandelion growing out of a crack in the sidewalk, that's a spontaneous plant. If you have a redbud tree planted in front of your house, that one is not spontaneous. Other things that are not spontaneous include things that are planted even in restoration areas. If they, will reproduce and then continue to grow, they can be considered spontaneous at that point. And also, there's something called a waif. So it's a single plant or population that they're not planted by humans, but they're not persisting. So, you have a seed that was stuck on someone's shoe that was able to get into the ground and grow, but after that one dies, then it's not still there anymore. So that would be considered a waif. So we had a bunch of data sources that were used to compile this information, including these two checklists, the one from Bob DeCandido et al. And one that Daniel Atha and Brian Boom put together in the beginning of the Ecoflora project. We also had the iNaturalist, thousands and thousands, tens of thousands of iNaturalist observations, some data on native plant occurrence from New York City parks that they kindly shared with me, herbarium specimens that were housed in New York Herbarium as well as other herbaria, and the very invaluable expert opinion of many people that are working on the ground in New York City every day, finding new, cool, and amazing plants. So that resulted in, this was contained both historic and current taxa. So there were over 3,000 species that came up combined in all of these. So we had to figure out which ones were indeed still here. And so I present to you the spontaneous flora of New York City with 1,262 taxa. Taxa basically just includes species and subspecies, because there are a few kind of subspecies that are also here. This includes a majority angiosperms, but also 14 gymnosperms and 33 ferns and lycophytes. We've got 64% native, which is in the same kind of range Mila had mentioned about, I think, 68% native species that they had found in their previous study. And this tracks with other large cities throughout the world. We also have quite a number of state endangered, threatened, and rare species, as well as one federally threatened species, which is sea beach amaranth, I believe. Some other statistics, there's 147 plant families represented. The most represented families are ones that might make sense. Poaceae, the grass family, Asteraceae, the sunflower family, Cyperaceae, sedges, Rosaceae, and Fabaceae, the pea family. You can see that they all have kind of different values of how many of them are native versus introduced, with Cyperaceae being the one that has the most proportion of native species, and Fabaceae with the most portion of introduced species. If we look at a genus level, we've got over 500 genera. Carex here is still the most represented, which contributed a lot to that Cyperaceae, as well as Cyperus. We've also got some large genera with almost all native species, Solidago, Juncus, and Quercus, Symphotricum, etc. Looking at some other statistics, plant habit, most of the things that we have are herbaceous, about almost exactly 50%, followed by graminoids, shrubs, trees, vines, ferns, and less than 2% of both aquatic and parasitic species. So we've got a great a variety of different kind of habits. Looking at the National Wetland Plant Ranking, this is going to classify things into whether they're a range from obligate wetland plants that always occur in wetlands to upland plants that almost never occur in wetlands. And when we look at that, we see the highest proportion that we have are facultative wetland or facultative upland plants. So they will almost always occur in upland, but they can occur in wetlands. If we look at just the native taxa, we see that the numbers go down, but the distribution is roughly the same. So we have the facultative upland followed by the obligate wetland and upland plants. And they're kind of evenly distributed almost. So going back in time, we're looking at the historical flora of New York City. Back in the 1800s, there was a lot different going on here. In the last 200 years, we've had a ton of growth that's happened in the city, lots of habitat changes, and that's can be seen in the numbers that we'll see. So we had these two sources that I mentioned from 1819 and 1915. I had to go through all of these, standardize all of the taxonomy, update the taxonomy, and determine what they had said was here. We end up with these numbers. So with a caveat here, the 1819 numbers are missing some native species, and I'm working on this, but we're going to say that it's more like that. So you can see that over time, the number of species has kind of changed slightly, but what's really happened is that the percent native versus introduced has changed. Over time, we've had a higher proportion of introduced species, which makes sense. And we're going to talk about a couple of these. I know I'm probably going to have to skip a bunch of things because I have way too much to talk about, but stay tuned because in a few months, I'll have a full-length presentation of this to be able to give. So these are all the traits that we looked at with these different categories to compare the habit duration, the photosynthetic pathway, leaf periodicity, the sexual system, pollination syndrome, spininess, clonality, wetland rating, and coefficient of conservation, which is how habitat specific there is. So if we look for these persistent natives, these are species that were present before 1915 and are also present now. That's about 700 taxa. Some native genera that have fully persisted that have had no species loss are oaks and smartweeds. Some other highly persistent genera include the ones that you see here, which contain a mix of habit types. We've got trees, grasses, and herbaceous plants. When we look at the coefficient of conservation, so this habitat specificity with the persistent natives, you can see that most of the ones that are persisting are in this range of like 4 to 7, which are intermediate ecological tolerance and slightly more narrow ecological tolerance. So these are things that are associated with stable community or slight anthropogenic disturbance. So that's really nice to see. When we look at the lost natives, there's slightly less taxa, about 500. The most common families with the native species lost are most of the generally large families as well. So Cyperaceae, Poaceae, and Asteraceae, but also orchids have seen a lot of species loss over the last 200 years. Some native genera that are completely extirpated include sundew, which formerly had 3 species present in the city, and the bluet, which had 2 species present in the city. Some other greatly reduced genera include Platanthera, which is an orchid, and Utricularia and Ptamogeton, which are both aquatic species. There's lots of reasons why these things might have been lost, including habitat loss, overharvesting, and competition from other species, as well as misidentification or misapplication of names that perhaps some of these were indeed never here in the first place. But it's hard to know that when someone has published something 200 years ago. When we look at the persistent versus the native taxa, there are a couple of these traits that significantly differ between these two groups, including there was a higher percentage of evergreens that have been lost, which mainly is due to evergreen ferns that were lost. A higher percentage of vines, graminoids, and trees are persisting as opposed to herbs and aquatics that were lost. A higher percentage of C4 plants persist, which is explained by some of these specific genera that have a lot of C4 plants. And also the wetland rating, a higher percentage of facultative upland plants have persisted, while a higher percentage of obligate plants are lost. And we look at some of these taxa, you can see kind of a spread of ones that have been completely lost, have been kind of evenly lost and persisted. And then on the other end are things that have greatly persisted. We look at the introductions. There are two interesting genera that, so Trifolium has 6 species that are present in New York City. 5 of those, they're all introduced. 5 of them were introduced before 1915 and are still found today. Similarly, in Veronica, the speedwell, there are 8 introduced species, 6 of which were introduced before 1915. Something interesting here as well, you can see them mentioning here in the 1800s and the 1900s about this mysterious plant, Cicimbrium alliaria or Alliaria alliaria, has started to grow, and they see no reason why it should not spread. This, of course, is the infamous garlic mustard, which now is found all over the city. So it's really interesting to look at these historic things to be able to track as these invasive or introduced plants have started to get a foothold here. Some other notable ones are Rubus phenoclassius, the wineberry, which was mentioned in 1915 as a rare garden escape, and Lesser celandine, which was listed in 1915 as a fugitive from Europe, known only in a few locations, which now, of course, is totally blanketing the Bronx up here in certain areas. The lost introductions, about 500 taxa. There's a lot of like strange things going on here. There's stuff from a lot of things from the South America, Southern Hemisphere that was probably introduced through shipping lanes, etc., that have since been lost and have not remained. And finally, we have the post-1915 introductions. So these are the more recent introductions. I wanted to see, are these different than the things that were introduced in the past? And indeed, we do see some things. A higher percentage of trees and shrubs have been introduced, higher percentage of evergreens, higher percentage of perennials, and interestingly, a lower percentage of clonally reproducing taxa. And instead, we have these more kind of like long-lived perennial woody species. And so the last kind of category that's kind of weird are these gained native taxa, because how can we gain native taxa if they're native? What's going on here are a couple things. We can have taxonomic changes, some things that are missing from the previous lists, or again, these kind of misidentification or misapplied names. There are some of these that have records of herbarium specimens that they are indeed listed as having been here, even though they're not listed in those sources that I used. So that's something that I still have to look at to kind of supplement with that. And Cybraceae is one of the largest kind of game native taxa. Okay, I'm sure I went over, but the point here, over the past 200 years, we've seen a great change in the landscape, the climate, and the makeup of our plant, animal, and human components. While much has been lost, much still remains to protect. And this information that we've compiled can be used in a lot of ways to inform current and future management practices, to illustrate to others the state of our spontaneous flora and the kind of positives that are here, and to help contextualize trends in species introductions and losses so that we can help plan for the future. And with that, we'll thank everybody that we worked with on Ecoflora and close it out.
Thank you very much, Lydia, for sharing information about the past and present native and naturalized flora of New York City. Really appreciate it. Excellent presentation. Next, we have a presentation by Dr. Christopher Terrell, who's a research curator of botany at the Milwaukee Public Museum and a self-employed software developer who has experience with several natural history data projects, including the development of the Symbiota portal for the Ecoflora of North America project. Chris received a bachelor's degree from the University of Wisconsin-Stevens Point, a master's degree from Iowa State University, and a PhD from the University of New Brunswick, self-described as a botanist by training and a hobbyist computer programmer. Dr. Terrell is going to talk about new features of the Ecoflora Symbiota portal. Unfortunately, Chris cannot be with us today, but this presentation has been pre-recorded, and Lydia will now share the recording on her screen.
Hello, everyone. My name is Chris Tyrell, and I'm sorry I can't be with you live this morning, but I'm going to share with you a little bit about what I've been doing with Ecoflora. So professionally, I am a botanist at the Milwaukee Public Museum in Wisconsin, and as a hobby, I do computer programming. And the Ecoflora project gave me the opportunity to kind of combine those two passions of mine, and that's what I'm going to present to you today. So let me just share my screen here. Right. So now, so some of the new features that are in the Ecoflora Symbiota portal. Now, let's step back a second. So this might be a review for a lot of you, or it might be new, but I just want to make sure we're all on the same page here. So places like the New York Botanical Garden has a lot of living collections, and people go there for, walking through the greenhouses and the collections. But maybe what you don't realize is there is an enormous museum of plants that is also on the site here. And these plant museums are called herbaria, or a herbarium. And herbaria are all over the world. New York happens to have one of the largest on Earth, something around in excess of 5 million plant specimens or so. And these museums of plants, they are actually plants that are pressed and dried and mounted onto archival pieces of paper. So here's an example of what I talk about. This is a herbarium specimen. This plant was collected and it was pressed, dried, and then glued onto this sheet of paper. And this simple process of pressing and drying the plant will keep it preserved for hundreds of years. Some of the oldest herbarium specimens are close to 500 years old now in Europe. And what's really, important about these specimens is you have the actual physical plant in a dried form, and so you can see all of the characteristics of the plant. You can look at the flowers if the flowers were collected. You can look at the leaves. You can look at the stems. In the last 30 to 40 years, we've been able to actually extract DNA out of specimens like this. So we could take a part of the leaf and crunch it up and extract and sequence the DNA from things. But really, one of the most, or probably the most critical portion of a herbarium specimen is the data label. And the data label is down in the lower right corner on this photograph, and it contains information about what plant that is, who collected it, when did they collect it, where did they collect it, and potentially some information about the environment or associated species that were around it. And if you start, aggregating 4 or 5 million specimens like this, you begin to get a picture of the distribution of plants. Where was a particular species found across space and also across time? And that's really, really important information. It serves as the basis of a lot of our natural resource management, interstate commerce, invasive species, all sorts of questions that we can ask and answer using just these herbarium specimens. These specimens serve as vouchers for things called checklists. And I'll get into that in just a second. But first, I want to mention that museum specimens are oftentimes served to the public on the internet using what we call a portal. And one piece of software for doing this is called Symbiota. And Symbiota, was developed about 25 years ago for doing just that, for serving museum specimen information to the general public. And Ecoflora has a Symbiota portal that is connected to all of the herbaria on the East Coast or in the Eastern United States. And so you can collect information or view information about the specimens that are in New York City that are related to the Ecoflora project or within the footprint of the Ecoflora project. And so if you're not familiar with that, you can check out that site. But if we look at one of these particular checklists, actually, let me take you to the web and we will look at this checklist here. So if we go here, we are on sorry, I have to reshare a different screen with you. Okay, so if we go here, now we are on CERNEC, the Ecoflora Symbiota portal. So remember, Symbiota is just a piece of software. It's the name of it. And the portal is a web-based platform where individuals, the general public, can go and look at museum specimens. So I'm going to click on this Ecoflora page. There's a number of checklists. And like I mentioned before, checklist is just sort of a list of all of the species that are growing in an area. And we can kind of take a look at this checklist. So we have here, it describes what the area, this is Central Park's checklist. It's part of the Ecoflora project. And if you scroll down, you can see a number of scientific names that are all plants that are from, putatively from Central Park. So the big bold name is the taxonomic family, sort of a high level organization. And then each species is listed underneath its appropriate family. So we can kind of look, if we look at the sweet gums here, we could click on a link and take a look. And these are now records of specimens of sweet gum that are in the herbarium at New York Botanical Garden that are also from Central Park. So you can see some information about those. You could click on a photograph to get more information. Now, another thing that a checklist will do is it will allow us to map all of the records that we associate with the checklist. So if I click on this map here, should be able to see, so now we have a layout of Central Park, and you're looking at a bunch of pins, and each pin represents a different museum specimen. So if I clicked on one of these, it'll pop up and show us information about who collected the specimen, where it was collected, when it was collected. All that label information is here in the portal for you to see. Now, these vouchers are, they're really important because if there's any question as to whether something was growing there, let's say someone reported seeing a really rare species in Central Park, and perhaps there was a development project or a infrastructure repair or something like that, it'd be important to know if there's going to be detriment to the environment. That's just one example. There could be many number of examples. It could be invasive species was reported in the park. And the thing is, if we don't have a voucher, if there's not a physical specimen of that, it's hard to confirm whether it truly was something that was invasive or rare, or if it was just sort of something that looked similar. But more and more, we've been using high quality, high resolution photographs and detailed photographs as vouchers instead of having a physical specimen. Physical specimens, they take up a lot of space. They're, expensive to store, and it also it removes a plant from the environment. And so photographs are what's the phrase I'm looking? They're a non-destructive way of sampling and vouchering plants. And so that's really one thing that iNaturalist is doing, is allowing for these non-destructive vouchers. So all of you, I'm sure, are familiar with iNaturalist. You've been posting observations to it. And those observations have photographs that serve as a voucher, because those photographs have who took the photo. The photo is the physical or the morphological representation of the plant. What plant was it? And where was it collected? So all of that label data is also available in iNaturalist. So what we did with Ecoflora is we wanted to merge those two. We wanted to take the iNaturalist records and the Symbiota herbarium records and kind of put them together in one place. So that's what I'm going to show you here. What you're seeing on the screen, this is a sort of my internal development version of the Central Park checklist. And you'll see it's very similar to what I showed you before. One thing that I can note is I can turn on something called the vouchers. And if you look in blue text, for various species, there is a voucher specimen. This is a herbarium specimen that represents that species in Central Park. So if I clicked on that, there'd be information about it. You can see here it was collected in Central Park. And that is a museum specimen that is vouchering that species as being in that place. Now, the first thing we had to do is we wanted to associate our checklist with an iNaturalist project. So in order to do that, I added some new features to the Symbiota portal that allows us to connect to an external service. So in this case, it's iNaturalist. And what we're going to do, we're just going to grab the name of the project, which is New York City Ecoflora, and add it into Symbiota. And because I'm a botanist, I'm going to limit this to just plants because they're the best. No. And so I save that. And now the checklist has been associated with our iNaturalist project. And if you watch the screen here, watch the checklist a little bit. In a minute or so, hopefully, we're going to see some icons pop up. So they just popped up. There's these little green iNaturalist bird icons that just popped up. And what's happening behind the scenes, the programming is actually reaching out to iNaturalist, and it's asking, it's saying, hey, I have this checklist of museum specimens. Here's all these species. What does New York City iNaturalist Ecoflora project have for each of those species? And when it matches, it will give you the little bird icon. So if you look, this Amaranthus powellii, there's no bird icon there, so it was not able to find that species in iNaturalist, even though there seems to be a museum specimen for it. And if we click on one of the birds, that will take us to iNaturalist, and we can see all of the observations of the sweet gum that is within our New York City Ecoflora project. So these are just the ones that are restricted to the New York City Ecoflora project. So that's kind of fun. So that's one addition we can do now that we've associated our iNaturalist project with Symbiota. Another thing we can do is we can map some of our stuff. So you see the red dots here, those are the museum specimens. And here in just a second, there's a little delay because it's got to reach out to iNaturalist. But the software has now reached out to iNaturalist and said, hey, give me back all of the records, the plant records you have that are for the New York City Ecoflora. And so the New York City Ecoflora is a huge area, and the map tries to restrict it down to the particular checklist. So in this case, it was Central Park. So we just have, you're seeing this small area, which is sort of the radius around Central Park. But if I look at one of these green dots here, if I mouse over it, it'll tell you a little information about it. But if I click on the dot or the pin, it will bring me to iNaturalist, and it will bring me to the exact plant or the exact observation that that point represents. So you can see here, it was Japanese lantern growing in Central Park. So that's pretty cool. We can now combine, you can see the red are the museum specimens and the green are the iNaturalist observations. So you can very quickly see which things are which, and really you can zoom in on them and get some information about each of these items. So there's a nice lobelia there. Okay, so that's the map. What else do we have? Oh, and now, remember how I mentioned we had those vouchers? The other thing we can do is we can add vouchers from iNaturalist. So I should note, when you're looking at a checklist on Symbiota, there's two roles that a person can have. A person can either be a general user, such as a member of the public, where they come to the checklist and just use it and view it and click on things. Or a person can be a manager of the checklist, which means they're more of an administrator. They may have set up the checklist, or they have higher permissions or privileges in order to edit the checklist. So you as a general user wouldn't be able to add vouchers to a checklist. Because I'm the manager of this checklist, I am able to add vouchers. So that the next thing you're going to see is something that you won't be able to do, but I'm just going to kind of demonstrate, if you were a checklist manager, how you would do this. So we click on the Add Voucher feature of Symbiota, and then there's a new link here that says Add Vouchers from an External Service. When I click on that, we see a new version of our checklist. And what the checklist has done, there is a white background in this box if it matches something in iNaturalist, and a gray background if there wasn't a match. And we can click on the name. So I'm going to do my sweet gum again. I'll click on the species name for sweet gum, and that's going to jump us to iNaturalist, and it's going to show us all of the records that are from the New York City Ecoflora that match that species. And if I want to just choose, I'm going to randomly choose this one. It's probably not in Central Park. Let's see if we can get one in Central Park here. Try this one. Okay, so yeah, it looks like a sweet gum. Now what I can do is there's a unique number that iNaturalist gives these observations. I can grab that number and go back to Symbiota. I can put the number in, and then I can ask Symbiota to get the information for that voucher. So you see it pulled up Tatiana Poliano, 2019, October 3rd. Let's just double check. This was collected by Tatiana Poliano on October 3rd, 2019. So it is getting, linking back to this record. And then I can save that voucher to the checklist. We go back and look at the checklist and I turn on the vouchers. Now what we're going to see is under our sweet gum right here, we actually have two vouchers now. We have the voucher that has this NY. That means it's from New York Botanical Garden. It's a museum specimen. Then we have this other voucher, the Tatiana one, that is the iNaturalist voucher. And if I click on the link for the iNaturalist voucher, it takes me right back to that same record, observation record. And if I click on the museum specimen voucher, it will pull up the information within the Symbiota portal about that museum specimen. So it looks a little terrible on my computer because I'm using a local development version. But if you do this on the web, on the actual portal, it'll look much better and much cleaner. So that is, in a quick nutshell, everything that I have been working on for the EcoFlora project. And I'm happy to take any questions. I will leave my email with all, in fact, let me just put this quick. My email is Tyrrell, Tyrrell @mpm.edu. You can send me some questions if you have any questions. I'll be happy to answer them. And thank you all for listening.
Thank you, Chris, for that recording your presentation. And thanks, Lydia, for sharing the recording. I really look forward to exploring all these new features of the Symbiota portal, which, because of these recent features added, now makes it even more powerful as a tool for learning about studying and conserving urban plants. Our next speaker is Dr. Eric Sanderson, who's Vice President for Urban Conservation Strategy at the New York Botanical Garden. Eric is the best-selling author of Mannahatta, A Natural History of New York City, and the critically acclaimed Terra Nova, The New World After Oil, Cars, and Suburbs. Eric has published extensively on issues of landscape planning, wildlife conservation, and urban resilience. He was also an invited speaker at the second annual EcoFlora conference back in 2019. We welcome Eric back for his 6th conference, now as an NYBG staff member. Well, and he's going to talk to us about Nature Your City, making New York City better for people and for plants. I turn it over to you, Eric.
Great. Thanks very much, Larry. Let me share my screen here. Okay, thank you very much. So I'm going to build on the presentations we've already had today and talk to you about the emerging urban conservation strategy at NYBG that builds strongly on the great work that Lydia and Brian and Tommy and all of you have done over the last years on the EcoFlora project. And I have to say, you know, this work in urban nature, I think it's growing in not only interest, but importance to the future of cities. I, like many of us, am really concerned about the way that climate change is going to impact the city that I love, New York City, and other cities around the world. And I really think that Nature Your City is the way that we get through the climate crisis here. Both trying to mitigate the climate crisis, but more importantly, adapting to the climate crisis. And I'll try and talk to you a little bit about what we're trying to do here at the garden. So why Nature Your City? This is a picture of the Botanical Garden grounds on June 7th of this year, when the smoke from the boreal forest wafted down, hundreds of miles into the city. And I think this kind of event leads to lots of conversation about what we need to do in the future. And really it's about what is the connection between nature and the city and vice versa. And traditionally, these two things have been thought to be in opposition to each other. But I think the emerging understanding that comes out of the research, like that Mila and Lydia were sharing, is that these two things are actually tightly connected to each other. And they're tightly connected in a number of different ways here in the 21st century. Nature literally enables cities. You can't have a city without nature, without the food and the water and all the ecosystem services that nature provides. Nature also makes cities more livable. You know, witness the experience of Central Park and the creation of Central Park and how that actually facilitated people being able to live in Manhattan in the 19th century and the kind of model that it's set for cities around the world. Nature confers resilience. Nature knows how to last because it's been here from the very beginning. It's very well adapted to our world and it continues to change with it. And I would argue that actually nature makes cities great. That, you know, New York City wouldn't have the particular kind of characteristics, its particular way of living here if it wasn't for the fact that it's an archipelago of islands in an estuary with that kind of ecosystem diversity that we've already heard about. What's less often said is that I actually think urban life is also the key to saving nature, not just in the city, but globally. And what I mean by that is that cities are more environmentally efficient in terms of the resources that are used per capita. And I'll show you a little bit of information about that in a second. They are space saving because people in cities live at higher densities than they do in rural environments, almost by definition. But I think we forget that that's actually a critical thing we need to do to be able to create space for farms and fields and nature preserves and wild places. Cities are also the place that ideas come from. I mean, not every idea comes from a city, but the concentration of people and the interactions between people actually promotes ideation and it creates wealth and power and it concentrates those in cities. And so I think if we get cities on the side of nature, then that'll make better nature. And if we have better nature, that will make our cities better. And so this is actually a kind of positive feedback loop that we're trying to create to fight the kind of other positive feedback loops that climate change is imposing upon us. And just to reiterate this point, this is a slide from the New York City Nature Goals Coalition, that everyone deserves great nature. Every person, every plant, every animal, we all deserve great nature. And I when I first moved to New York City, I was, so taken by the, by the ambition that New Yorkers have, for their economic lives and for their cultural lives and for their artistic lives and indeed amongst the scientists for our, scientific endeavors. It just seems like there is no limit to what we can learn and what we can know and the way we can experience this world. And yet people were so, pessimistic about the nature that we can have. It's like you had to make this trade if you wanted to have, the Yankees and the Mets and, a good paying job. You necessarily had to live in a polluted and depauperate place. And I actually think that's just, it's horseshit, if you use the expression. It's just the wrong idea. Like, we need to bring that same level of ambition to the nature of our city and to the nature of our world if we want to make a difference for the future. And actually, I think that younger people, people of the, if you like, the Gen Z generation, people of my son's generation, they believe that is true, that you can have a great job, you can, go to the opera on Friday night and you can be kayaking the next morning. And that is just what it means to be a New Yorker. And to sort of bring a little bit of data at this point, it's true that if you compare the average American lifestyle to the average New Yorker lifestyle, that the average New Yorker on a per person basis, not in aggregate, but in per person basis, uses 74% less water per person and 35% less electricity and 45% less garbage. And this point was brought home to me in sort of 2008, 2009, when the city was working on its first sustainability plan, Plan NYC. And I just, I think this is such a striking point, and you see it across all kinds of metrics now. Carbon dioxide emissions, for example, are so much lower in denser situations for a variety of related but kind of different sorts of reasons. Recently, I've been adding this slide to my talk, which is that if you look at the 50 states in the United States, all these states in orange have fewer people than New York City does today. Just think about that. There are more people living in New York City than in Colorado, right? Or in Wisconsin, or in South Carolina. I think there's some political implications there that I won't draw out, but I'm sure you can. but also this idea that it's so space saving, that it's so compactful. And that is actually creating more space for plants elsewhere by us living in a concentrated way in New York City. So what is Nature Your City going to do here at the Botanical Garden? It really has 4 programs of work that I kind of think of in the way that the Lenape people used to talk about the Three Sisters Garden, which is that, Lenape used to build these mounds in the springtime. And for us, that mound is the historical ecology of New York City that I've given 25 years of my life to. So understanding in detail the way nature wants this landscape to work, and to use historical maps and references and some ecological modeling techniques to try and project backwards about what that design that was in the case in say 1609, say when Europeans first arrived. Not to say that it never changed, but to understand the dynamics of that landscape before European intervention. And then from the historical ecology grows ecological democracy. So this is a program of work around Vision Maker, a tool for allowing everybody to plan the ecological functions and ecosystem services of the city. Active restoration work. So this is leveraging NYBG's really deep experience and skills in practicing horticulture and trying to use those horticultural skills to actively restore places around the city. And I'll talk about that in a second. And then this third piece, which is the environmental governance piece. So helping New York City and other cities around the world to establish the kinds of, legal and social and norm-based frameworks that encourage us to bring more nature into our cities. So I'll just briefly speak about each of these 4 points. So many of you know about my work on the historical ecology of Manhattan, and we've been expanding that work over all of New York City. And I'm working on a book now that'll come out in the fall of 2025 that will describe the original topography, the original hydrology, the original shoreline, what the Native Americans were doing, and what happened to all these features. What happened to the hundreds of miles of streams and the dozens and dozens of springs and hills and the 540 Lenape sites. And the reason we do this work, the reason we explore the past, is because it becomes a guide for the future. It tells us, nature's version of the landscape that became New York, and the dynamics that made that happen, the dynamics that are not just of the year of 1609, but the things that happened, in the centuries before that, and since the glaciation, and since the bedrock was created, millions and millions of years ago. I also have found, that talking about the Lenape people and understanding the way that people have lived in this landscape over the last 8,000 years before colonization is a really powerful way to talk about a different set of values and a different set of practices. Not that we want to, adopt all of those, full cloth, but in fact that there's many ways of living in the New York City landscape. And finally, I think there's such a great value in appreciating the amazing diversity and productivity of this place, which is actually the diversity and productivity that made New York City possible. Mila made this really important point in her presentation that cities tend to be established in places of high biodiversity potential. And, through a very careful and rigorous examination of the historical ecology of New York City, we can provide thousands of data points that demonstrate why that is the case. And we do this, as I mentioned, through historical maps, through taking maps of the city. When I was working on Manhattan, there was basically this one map, this British headquarters map that's at the top here. But as we've worked on the rest of the city, it's been hundreds of maps from different points in time, which need to be read as historical documents, made for particular reasons by particular people or particular technologies, interpreted in the way historians look at these documents. But then trying to derive from them what we can about the ecology of the place, about its geomorphology and its soils and its geologic conditions and its ecological and its biodiversity. So then we take all these things that we, my students and I, this has been a project that hundreds of people have contributed to over the last couple of decades. We digitize the features. We put them together to make maps of the topography and the hydrology and so forth. And then we connect those using these mirror webs of ecological relationships that try to infer the distributions of plants and animals. That's what underlies these sort of digital viewing points, which we're in the process of updating now with the citywide data, where you'll be able to zoom into a particular block in the city and get a list of all the plants and animals that used to live in that block. And I really think this is the kind of, context setting sort of work that then, helps us understand the work that, Lydia is doing about what we've lost, over the last 400 years and what we can bring back potentially. And, Mila's work about understanding the distribution of species, in cities around the world and how that's changing and how we can make the lives better for other critters. I'm working on this atlas and gazetteer, which will have very much larger scale maps than in Mannahatta of the historical landscape across all 5 boroughs, and then writing these descriptions of about 3,000. There's about 3,500 maybe, but anyway, many different features, some longer, some shorter, but trying to tell the story about the lakes and the springs and the hills and so forth. The trees in some cases where we have individual trees, and describe what they were, why they were called what they were, how they related to the landscape where they were found, and then what happened to them. Through our work we go through a lot of old resources, old maps and local histories and so forth that are not otherwise very well known. So these lines at the bottom are very important to me, listing the text, the documents, and the page numbers and the maps. And these are not meant to be exhaustive resources, but they're meant to say, if you really want to know more about this place, start with these resources and then take off on your own investigations. So that's the historical ecology foundation that we're building here. The second part is about ecological democracy, and this is trying to put the tools in the hands of New Yorkers to imagine a better ecology for their own neighborhoods, and then to be able to share that with other people. So we have this platform that I built when I was at WCS called visionmaker.nyc, again, with contributions from very, very many people, and including some people that are on the call today. And we want to launch VisionMaker 2.0 that builds on the experience of what we've learned about VisionMaker over the last 10 years. So this idea of ecological democracy is that if you really want to change the environment, if you really want to make the environment better for people and for nature, you need to involve people from the get-go. And so VisionMaker is a tool to do that. It's a computer-based tool, it's an internet-based tool, it's free. And what I find about it is that there's this, interesting conversations that come around VisionMaker. It's not just about using the technology, it's actually about the conversations that surround the technology about, well, what do we really want? And what can we really do? And what are the sort of basic assumptions that we're all bringing to our neighborhoods and our lives in the city? And briefly, the way VisionMaker works is you can see a map of the city, you can see the historical ecology of the city, you can see the current ecology in terms of a map of about 75 different ecosystem types and then modifiers of those ecosystem types. You can zoom into your particular neighborhood. This is Mill Basin in Brooklyn. You can see where that sits in the historical ecology. Sadly, Mill Basin is mostly on salt marshes. And then you can remap it. You can actually change the ecosystem distributions. You can select some parameters around the lifestyle of the people that live in the city, of your idea of the city of the future, and you can change the climate scenarios. And then that generates a set of inputs to models that then run in the background about the water cycle. I'll tell you about it in a second, that show you how your place relates to today and 1609, and what your vision is of the future, and what it means in terms of the water cycle, the carbon cycle, biodiversity, population, economics. And we're trying to add in some new things like urban heat island effects and flooding from intense storms and so forth, so that it becomes more and more useful to each of us. Each of us as individuals, each of us as sort of community activists, but also to the kind of professional set, to the architects and the landscape architects who have some tools for this sort of thing, but nothing that really goes in depth to the ecosystem ecology of New York City like VisionMaker does. And then, of course, then once you get the vision you like, you can then share that vision. You're not required to in any way. You can keep your vision as private for as long as you want, or you can just share it with a group of people that you curate, or you can share it with anybody and everybody on the web. And the reason we do this is because we want to involve people in designing the future by putting more plants in the ground using the horticultural resources and the scientific resources of the New York Botanical Garden. And this is, you know, there are many kinds of green spaces in the city. There are community gardens, and NYBG has this really strong Bronx Green Up program that is working all the time with community gardeners to help them, you know, to cultivate the soil and grow the plants they want. Those places also provide biodiversity benefits and biodiversity services and climate resilience services. So how can we amplify that? How can we help think about these bioswales that the DEP is putting in all around the city, or the green roofs that, you know, private individuals are putting in? You know, often these are very small patches. As we all know, as we walk around the city, you know, some are better maintained than others. So how can we, you know, help promote the horticultural skills and the interest and the value of these things so that they continue to do their ecosystem work? A part of this is also recognizing places in the city that are still flooding. Like this is a little piece of Casino Park that floods. It turns out this is also a place that used to be a stream, so it's no wonder that it wants to flood. But it's right next to a neighborhood that floods on a regular basis. So how can we, you know, use these kind of spaces to make them more climate resilient while still doing all the other work that, you know, parks need to do, provide biodiversity, provide playing fields, and provide recreational spaces. And then finally, you know, to see how this all fits together. You know, the city is actually changing all the time. You know, just like nature is changing all the time, the urban landscape is changing all the time. The problem with the urban landscape is it's sort of so finely divided into individual pieces of land that are each have their own, you know, property manager, their own owner. And, you know, in the United States and our public-private, you know, in our capitalistic system, those landowners have a lot of say about what goes on the individual pieces. So we're getting all these incremental, you know, improvements in general, sometimes not improvements, but, you know, all these incremental decisions are getting made. And the only thing that sort of knits it together is, you know, the law, you know, the zoning code on the one hand. But more importantly, I think there are these norms of behavior and these values. What do we value about how we live on the landscape? And I really think that's the thing that's changed, you know, say, over the last 50 years in New York City is, you know, I think in the past, people thought nature wasn't that important and it was the thing to be used and abused, but not so important. And I really think there's been a revolution in our values that we bring. And it's just in time because climate change is coming to get us, right? With more intense rainstorms and higher levels of sea level and hotter temperatures. And nature is going to be such an important part of the way we address adaptation in the city. And that's why the environmental governance piece is so important, trying to connect nature to the decision-making processes of the city. And here I'll just highlight some work that we've been doing with the Department of Environmental Protection over the last couple of years. You know, if you read the literature on climate resilience about New York City, it's getting more and more worried over time, and realizing kind of the immensity of the problem that we all face. The crisis literally that we face, and the necessity of dealing with that at the landscape scale, where, you know, just replacing all the sewer pipes is not going to be sufficient, and in fact would be so expensive and so disruptive that in itself could destroy the city that we love. So how do we think about this question? Well, what we've been trying to do is think about the historical ecology. What does it tell us about the places that used to flood? These are, you know, wetlands, ponds, streams, beaches, and the ocean, because, you know, we've had a lot of landfill in New York City. Places that were the ocean are now places where people live. Looking at the patterns of present flooding from 311 calls and other data sets, and then looking at patterns of future flooding from some modeling work that the DEP has done and then some modeling work that other people have done to say where the future flooding might happen. And then if you lay all three of those things on top of each other, the past, present, and future flooding, you get maps that look like this that are about 20% of the city, a little bit less than 20% of the city, about 1.2 million people live in these colored areas. And you can characterize the kind of flooding that will be most impactful, whether it's coastal flooding, you know, from big storm surges, a la Hurricane Sandy, pluvial flooding, which is, you know, from intense rain events, a la Hurricane Ida, or a combination of the two, which is actually the thing I'm most worried about. You know, like I said, Sandy was mostly a coastal storm without a lot of rain. Ida was mostly a rainstorm without a lot of, wind and coastal storm surge. But it's very easy to imagine you could have both those things at the same time. And that's the situation, particularly in southern Brooklyn, a lot of the coastal plain and in Queens and parts of Staten Island. That's going to be really, really very dangerous for New York City. So just to wrap up then, our Nature Your City program has these 4 pillars, these 4 programmatic elements. The historical ecology feeds the ecological democracy, the active restoration, the environmental governance. The environmental governance, the active and ecological democracy are meant to support the active restoration efforts. And of course, there's a crosstalk between those, because the more we build a constituency of people that care about nature in New York City, the more the politicians will listen to us and they'll be more receptive. Not that they're not receptive now, but I think there's still much more work to be done in this area to build really true and strong environmental governance in the city that'll be better for plants. And in this way, we create the future ecology of the city. Because, the city depends on nature, and the nature depends on the city. And I really think that the crisis that the climate change presents is the next major transformative process for New York City and probably cities around the world. And nature is the way. Nature will save the city and many others. And I'm just so glad to say that the New York Botanical Garden wants to be part of that process. So thank you very much.
Thanks, Eric, for sharing information on how we can make New York City better for people and plants. And I really appreciate the connection you made to the other presentations and to data on plant diversity. Really appreciate it.
Sure.
Our final speaker is Clara Holmes, who's a plant ecologist with New York City Parks Division of Environment and Planning. Clara has more than a decade of experience in plant community monitoring and conservation, restoration planning, and project management. At New York City Parks, Clara monitors salt marshes, tracks rare and locally sensitive plant species, and reviews planting palettes for restoration and construction projects. Before working for NYC Parks, Clara managed seed collection projects for the Mid-Atlantic Regional Seed Bank. She has a master's degree from Pace University, a bachelor's degree from the College of Charleston. Clara is also an alumna speaker from the second annual EcoFlora conference from 2019. Happy to welcome her back today to talk to us about sowing seeds, inspired projects helping to ensure a diverse flora. Clara?
Thank you so much, Lawrence. Good morning, everybody. It's really great to be back here at the 6th annual EcoFlora conference. And I'd really like to thank Lydia, Tommy, Brian, and others who have been working on the EcoFlora project for these past 6 years. I really think it has significantly contributed to efforts to reverse plant blindness. Excuse me, that more New Yorkers are seeing plants in new ways than ever before. And the recent expansion of the project to other cities has really extended that reach. I've long felt that the more people actually see plants, the greater likelihood that the diversity of the flora of New York and the United States can be sustained. It's my privilege and great pleasure today to present to you a few projects from New York City Parks that the Division of Environment and Planning is involved in that I believe are helping ensure the flora of New York City remains diverse for many years to come. These are projects that were started by several of my colleagues, and they would not be possible without the support and hard work of many individuals in the Parks Department, but also many of you community members out there. So first up is the development of the Pollinator Place Gardens. This is an initiative that was spearheaded by our Director of Horticulture, Matthew Morrow, back in 2019. And the vision, which really developed between Matthew and our former Deputy Commissioner, Liam Kavanaugh, is to create garden spaces that serve ecological functions and help inspire and educate park visitors. I think that making our gardens more than spaces that are just aesthetically pleasing to humans is critical in ensuring a rich floral and faunal diversity across the city. And really all it takes is making a conscious effort about what plant species go into these garden spaces. So to see this vision become a reality, the horticulture team developed a set of planting parameters that would help meet the gardeners where they are and also encourage them to be more intentional about their plant choices. Gardeners that choose to follow these parameters have their spaces recognized as a pollinator place with that sign in the previous slide. And these parameters, which you can see here on the screen, stipulate that in order for a garden to be considered a pollinator place, at least 60% of the plants need to be made up of non-cultivar plant species that are native to the New York City area. Gardeners are still allowed to use some amount of cultivars and plants that did not evolve in the region, but those are to be kept to a minimum. To help gardeners select ecologically valuable plant species, the horticulture team put together a plant selection tool that includes detailed information on life cycles, ecosystem services, the wetland indicator status, so on and so forth. They also put together a document that would help gardeners understand maintenance requirements for specific species. And these resources are really essential tools that can allow the gardeners to plan and implement the vision of ecologically beneficial gardeners across the city. So the first pollinator places were planted in 2020, and at that time there was one in each borough. And immediately the demand for these kinds of gardens was really high, and the program was quickly expanded to include conservancies, friends of groups, other kinds of partner organizations that manage gardens across New York City parkland. And as of 2023, there are 18 pollinator place gardens across the city. This year, the horticulture team thought it was important to do a first assessment of these gardens to understand how successful they are and what their needs might be. So these gardens were assessed, and generally the horticulture team came to the conclusion that they're all being very well maintained and that the plant health is very good. Of course, these gardens need a significant amount of maintenance, and in order for us to grow the program beyond these 18 gardens, additional resources are vital. Going forward, there is a desire from gardeners and from the horticultural group to diversify, first to expand and include more gardens, to diversify the species that are planted, and particularly to try to find a way to assess the insects that are using these gardens as a way to help qualify the benefits that the gardens are providing. Ultimately, I was really excited to be able to share this project with all of you, because the philosophy of these places is really spectacular. It's a beautiful concept. And I really think that the more it is implemented, the closer we get to creating these ecological corridors, these connections that Mila mentioned for wildlife and plant species across the city. So a big thank you to Matthew, the horticulture team, and all of the gardeners that have taken the initiative to create and maintain those spaces. And I have to be able to have a moment to show you a few of the great gardens that are pollinator places in New York City. So Damati Playground. You can really see the plant species richness here. Oops. All right. Next up is an initiative out of our Greenbelt Native Plant Center. It's an effort to develop a strong and successful planting palette for tree beds. These are extremely tough environments, and they exist all across the city. New York City has spent years successfully planting trees along our streets, and each tree has a certain amount of space, some more than others. And if we can find a way to turn these environments, we can and we can find a suite of species that consistently do well in them, we can create additional habitat, again, expanding those corridors for wildlife and increasing the diversity of plants in our city landscape. So though this project was led and coordinated by Patrick Over and others at the Greenbelt Native Plant Center, it's really individual New Yorkers who are implementing it. The concept's pretty simple. The Greenbelt Native Plant Center grows and provides plants to community members that are willing to plant, maintain, and collect data on the health and survival of species. Last year, Patrick and Sophie Plew developed a list of species that they believed would do well in these tough spaces and worked with a researcher in Chicago to develop a data collection tool that would be accessible to community members. In the spring of 2023, GNPC distributed plant kits and trained participants on how to collect the data. The kits included plants, tags for what species they received, a guide to the biology and ecological characteristics of the species, and a data entry guide. Participants are asked to submit observations once a month using an online form. And these observations include things on include information on the health and mortality of the plants, the care that they have given the plants, and the reproductive status, as well as a few other parameters. You can see here some examples of the codes that are used in order for individual New Yorkers to help contribute to our ability to develop a strong, successful species palette for these tough habitats. So 2023 really was the first year for this project. There are already 40 participants that have planted a total of 93 street beds, tree pits, and they're in all 5 boroughs. Nearly 4,000 plants representing 60 different species have been planted in these spaces, and over 1,200 observations have been made by community members to help us determine the right species for these spaces. So we do plan to continue the project in the future, but the initial results, of course, show that some species are doing better than others. For example, our narrow-leaf mountain mint and smooth blue aster are both generally successful in our tree pits, but surprisingly, some of our grasses, like little blue stem, is not functioning and is not as hardy in this environment as we might have expected. So it's been an incredible undertaking to coordinate this project, and a tremendous shout out to everybody at the Greenbelt Native Plant Center that has made this possible. And of course, thanks to all of you who are participating in the efforts to maintain and track these plants. We do hope to continue the project over the next few years, and that soon we will have a solid understanding of plant species that are suited to these tree beds so that everybody can turn their front sidewalk into habitat. The Greenbelt Native Plant Center is also involved in one of the next projects I'd like to talk about. For several years now, they have been producing seed instead, and not just plants, that can be used in restoration projects across the city. Increasing the availability of locally adapted seed is a national challenge, and New York City Parks is way ahead of other municipalities in helping solve that problem. But traditionally, the seed production efforts at Greenbelt have been focused on sort of our restoration workhorse species, those species that we need in really large quantities in order to do sort of landscape scale restorations. But over the last couple of years, we've incorporated one species that really doesn't fall in that category, and that's yellow thistle, Cirsium hiridulum. It's a locally sensitive species, and we only have 3 known populations of this plant across the city. And because it's a thistle, or being a thistle, it provides tremendous resources to a lot of important pollinators that call New York City home. And so realizing that one of our populations was likely going to be extirpated in the coming years, and understanding that the species holds valuable conservation and restoration potential, we embarked on an effort to collect seed of Cirsium hiridulum and grow it out in a seed production plot to increase our ability to incorporate the species into coastal restoration projects going forward. So in 2021, we collected and propagated seed from the species. And in 2022, 160 basal rosettes were planted into a seed production plot at the Native Plant Center. And this summer, we harvested 160 grams of that seed that will be used to grow plants for restoration projects over the next couple of years. And as a testament to just how valuable the species is to our pollinator diversity, not only is it pollinated and visited regularly by the common bumblebee, which you see in the upper right, but Greenbelt Native Plant Center Director Lucy Rubino observed the globally vulnerable golden northern bumblebee using the flowers in the seed production plot this summer. And you can see that photo there in the bottom of this slide. So we really hope that this model of using a conservation species for seed in a seed production plot can be a useful model for us to increase the species and diversity and availability for some of our restoration projects. And finally, I'd like to highlight new ways that New York City Parks is working to engage New Yorkers in the conservation of plant species. We have an incredible stewardship program and team at the New York City Department of Parks. And we have long been engaging community members to help care for our natural areas. And this year we worked closely with several super stewards and are continuing to work closely with these super stewards to help us manage a few rare plants. One example of that is on Staten Island, where we have a small population of Pygnanthemum verticillum, the world mountain mint, which is a state rare plant. And the population had been declining for several years due to growth of species like bedstraw and moth mullen. And this past spring and summer, we had a couple of very dedicated stewards who helped us remove those unwanted plants, and the number of stems of mountain mint has really recovered and expanded in that area. So it's been a really successful effort in a way to help us manage some of our conservation species and engage community members in new ways. Similarly, this fall, we'll be working with some super stewards to plant Amelanchier nantucketensis, the Nantucket Juneberry in Kaiser and Marine Park. This is a species that we don't have very many populations left in New York City anymore, and it's been challenging for us to replant and restore and reintroduce these kinds of sensitive species because they usually need extra care that we can't provide to every single one of our restoration projects. And so by engaging community members to help us plant some of these and provide additional afterwatering and additional weeding and care around these plants, we hope that these reintroductions will be successful. So we'll be working with volunteers to plant the Nantucket Juneberry this fall. So as some of the other presenters have mentioned, urban areas really are increasingly seen as crucial to slowing the biodiversity loss that's occurring across the globe. And ensuring that New York City has a diverse and ecologically rich flora is important for those efforts. And these projects I highlighted really are just a few. As you all know, we only had so much time to tell you all about the different work that we've been doing. And so these are just a few of the many projects that the Parks Department has undertaken to increase the diversity of the flora in our natural areas and in our garden spaces. And it would not be possible without the initiative of so many of my colleagues here and without help from the community members. I'm so fortunate to work with these people and with all of you New Yorkers who also care so much about the flora of New York City. So thanks to all of my colleagues and thanks to all of you for making this work possible.
Thank you very much, Clara, for sharing information about these projects. It's just great news and gives us all hope for a diverse urban flora in the future. And I'd really like to thank all of our presenters for so much wonderful information shared. We've had a lot of really excellent questions from the audience. So we're running short on time. We don't have much time for questions. I'm going to just limit to a quick question for everybody, but I will point out that many good questions in the Q&A, and many of these questions have already been addressed or answered by the panelists. So if you asked a question in the Q&A, please make sure to check to see whether it was answered. And I mean, I think I would, and there's lots. The questions range, great questions, lots ranging from details of an individual species to big systematic. Strategic approaches to how we address all of these issues that we've heard people talking about. But I think in wrapping it up, I would just say again, wonderful presentations. Thank you to the panelists. Here we have an audience that's a very engaged, informed, activist, committed audience. And I would really, so many of you gave so much wonderful information about how to make a difference and what we can all do and how the projects that you work on and the people you work with are making a difference. I would just like to go around and ask each person to sort of condense it down to a takeaway. What would you suggest for the person who says, what can I do in my daily life, or how can I make a difference for plant diversity, for the future of plant diversity, and to address the environmental challenges we face? Let me start with Mila. We'll go in order of the presenters.
Excellent.
Great. Okay. Thank you. Well, I think as individuals, probably one of the most important things we can do is to utilize native plants in our yards and gardens, and participate in some of these initiatives that Clara was just discussing in terms of nature gardening or nature pollinator gardens, and also the tree beds. And so these activities that kind of learning about native plants, but also promoting them in areas where people can see them, right? So our yards and gardens, our neighborhoods, our tree beds, our parks, and so on.
Excellent. Thank you very much. How about you, Lydia?
Well, I would say in the vein of Ecoflora is kind of educating people about what's here and why it matters, because I think especially when we're really entrenched in this kind of, we know what's here, but there's lots and lots of people that is really not obvious to, and I think the more people that know that everybody is a stakeholder in the current and the future, that will, I think, really inspire people to care more, even if it's just for the reason that it's it'll cost them money, or it's going to make people more uncomfortable or something. Like anything that can kind of motivate people, I think, is good.
Thank you. That's excellent. Coming from the New York Botanical Garden, we have a platform to do exactly that, and that we'll continue to do. Eric?
Yeah, I think I would say, try to think beyond yourself. I mean, I know we all try and do that, but cities have such important legacies of decisions that were made in past generations, and most of those decisions were made trying to solve some problem that generation had at that point in time. But often it turns out their solutions at that point in time create problems for future generations. And I really think we need to be more open and more giving to the future and trying to make decisions in our daily lives, in our neighborhoods, and as a city as a whole that give to the future and don't take away from the future. I mean, for many of us, I feel like we're feeling like society has kicked the can on these environmental problems for so long, and now suddenly we're holding all these cans, right? Like we're buried by them. And I just think, we not only need to find a way to deal with all that stuff that's been given to us, but we need to be offering solutions to the future. And that's actually why I think about nature restoration is so very important, because I really think it is that kind of thing that gives to the future in a way that future generations will really appreciate.
Good, thank you. Clara?
Yeah, so I'll echo sort of what Milo was saying, is that for those of you who have spaces to plant, to first be very intentional about the plants you choose. What plants get planted matters, and with the goal being something beyond just it being pleasing to our eyes. And then secondly, that for those of you who don't have those spaces, find our stewardship page. We have a really, really great stewardship program where we involve community members in planting trees and shrubs and some herbaceous material in our in our natural areas. And as I explained, we're expanding some of those opportunities beyond just planting and removing unwanted plants. So getting involved in your local garden, if you're really interested and have the time, getting training to become one of our super stewards, so you can sort of have the ability to manage a certain section of habitat with our guidance, and just understanding that it can be really overwhelming for people to get out in nature. And once they start learning more and more how to see plants and realizing that we are struggling in a lot of ways to maintain this floral diversity across the city, to find presentations like those that were given today and find more resources that can help encourage you in those moments of doubt, because it's not all negative, and we have to remind folks that we have to keep chipping away at it, one plant at a time. Otherwise, we wouldn't ever get there.
Thanks a lot, Clara. Great way to finish it up. And you're right, there's so much positive, and so much of you did a wonderful job sharing the positive and making us all optimistic and understanding what our role can be and how we can do much better moving forward. So we really appreciate this. This concludes today's EcoFlora conference. To the audience, I would say if you missed any part of today's event or want to watch it again, a recording has been saved and it will be published in the NYBG lecture library. Actually, a pitch for other programs at the garden. Please join us in person next week, next Thursday at NYBG for our Climate Week 2023 symposium, Cultivating Resilience. We're dropping a registration link in the chat so you can get more information about that. Incidentally, we'll leave the chat up for a few more minutes after we close out the program here, so you can look through the questions and gather information from the links we posted. On behalf of the EcoFlora team, I would like to thank all of our speakers and the audience participants for their excellent contributions to today's conference. Please follow links for more information on documenting plants, fungi, and their ecological partners in New York City. We hope that you are inspired to continue or to begin documenting, learning about, and conserving plants and their ecological partners in New York City or wherever you live. I want to express our deep appreciation for the Institute of Museum and Library Services for their generous support of the EcoFlora concept. And I also want to thank our partner gardens and the administrations of those partner gardens in this enduring endeavor to, along with vital governmental partners such as New York City Parks, New York State Parks, and most of all to the community scientists out there, past, present, and future, who sustain the project's vision. I'll close the conference with a quote from the late, great Professor Edward O. Wilson, former Distinguished Counselor of NYBG's Board of Trustees, who in 2016 wrote this about the New York City, about NYBG's EcoFlora project. As I wrote in my most recent book, Half Earth, like it or not, and prepared or not, we are the mind and stewards of the living world. The New York City EcoFlora project will not only prepare the city's residents to be good biodiversity stewards of their green metropolis, but will also inspire them to embrace such responsibility. With those words of inspiration and obligation from E.O. Wilson, I'll say goodbye for now. Please stay well, stay green, and take care.